Well, it's a real pleasure to begin and host the Office of Data Science Strategies seminar for data use and data reuse. Each month we take the opportunity to host a different institute from NIH and to highlight not only the work that the institute is doing in data science and data sharing, but also to highlight a particular principal investigator or awardee or even an intramural investigator and their research. So here you can see the plug for my office and the work that we do to create a modernized and integrated fair biomedical data ecosystem. So next slide, please. So again, if you're new to this format, please ask questions and let us know about yourself in the Slido. It's Data Science, NIH Data Sem 8 for this the seminar today. Okay, and just a reminder, the next seminar will be on October 15th. Next slide. So it's a real pleasure to introduce to you today my dear friend and colleague, Dr. Ishwar Chandraramaswamy. He is the Deputy Director of the Office of Data Science and Emerging Technologies, ODSET at NIAID, the Institute of Allergy and Infectious Diseases. Prior to this appointment, he was a program officer of bioinformatics in the Office of Genomics and Advanced Technologies, again at NIAID. And he also was the Associate Director of Cancer Biology and Genomics at the National Cancer Institute. And prior to that, Ishwar spent a number of years in industry. So thank you, Ishwar, for highlighting today the work that is happening at NIAID in data science and emerging technologies. And I'm going to hand it over to you.
Thank you, Susan, for the introduction and the opportunity for us to showcase some of the fascinating work happening at our institute. So the office is a relatively new office, and we have a new director, Wilbert Panhouse, who's recently joined us. And I serve as the deputy and have been so for the last about 3 years or so as we stood up the office. So moving on to the next slide. The office itself focuses and has the objective to build partnerships across the institute to harness the power of data in immune-mediated and infectious disease research. And it's a three-pronged approach by developing a robust research program, by providing data management and sharing guidance and implementation for NIAID conducted and funded research, and supporting programs in training and workforce development in a manner that aligns with the larger NIH strategic plan for data science. Our office itself is situated within the Office of the Director at NIAID. Moving on to the next slide. I wanted to just kind of put a plug in and showcase our Emerging Leaders in Data Science Fellowship program, which is in its 5th year now, and its focus is to build capacity and promote data science as a discipline in itself and train the next generation of data scientists. And moving on to the next animation here on the slide. The program itself is a cohort-based 2-year program where fellows conduct transdisciplinary rotational projects. And moving on to the next, you can move the animation, where they build technical and leadership competencies. We have 6 fellows currently, with 5 new fellows coming on board. And if you hit next, we have the next cohort and applications for which open in October next month. So for those that are interested. In the next slide, I'll take 1 minute to just showcase that we have a variety of trans-NIAID initiatives that focus on reusing data and secondary analysis of data sets generated from the breadth of programs across the institute. Programs that focus on software development, both investigator-initiated, driven by critical needs, as well as small business innovation research that with topics focused on data science. And last but certainly not the least, we have a contract award and a program to establish a federated data ecosystem, which is relatively a new award that was recently made and awarded to Seven Bridges and the Scripps Research Institute. If you hit next. So the objective of all of these programs and the efforts are really towards discovering and accessing data generated and the software developed from across NIAID funded and conducted projects. You can learn more about it from our website, and you can also follow us on our Twitter handle at NIAID. And in the next slide, I will, without further ado, introduce, and I'm delighted to introduce our speaker for today's data sharing and reuse seminar, Dr. Purvesh Khatri. He is electronics and communications engineer by training, has experience and expertise in software development, and has applied his transdisciplinary skills and serves as an associate professor in computational systems immunology. His research focuses on, if you hit next, focuses on developing machine learning methods for leveraging biological, clinical, and technical heterogeneity in publicly available data sets for accelerating translation medicine. And his lab has a dual affiliation with both the Institute for Immunity, Transplantation, and Infection, as well as with the Division of Biomedical Informatics Research in the Department of Medicine at Stanford University. The lab itself focuses on developing computational frameworks to integrate independent cohorts and better understand the human immune system, and develop diagnostic applications and tools to identify drug targets for inflammatory diseases. So with that, the title of his topic, and hit next, is Adventures of a Data Parasite, Accelerating Clinical Translation Using Heterogeneity in Public Data Sets. So I'll pass it on to you, Purvesh.
Thank you, Ishwar. I'm trying to share my screen, and I can't see the button that was there before for sharing screen now. Erin, any chance
Do you
can
see
help
it now?
me? No, I just don't see the button to share.
I made you the presenter. Do you see from the top where you can hit share?
The menu bar on top.
Well, maybe let me see, share content. Okay, I got it. And yeah, can you see my slide?
Perfect.
Yes.
All right. Thank you, Ishwar, NIAID, and Susan for inviting me to give the talk and share some of the work that we've been doing. Now, as Ishwar said, I'm an electronics and communications engineer by training. So I am not a biologist, and everything that you'll see today is only using publicly available data that we are using for translational medicine in inflammatory conditions. And just to drive home that point, this is my disclosure that I've never held or bought a pipette. This is the lab when we were moving the night before we were moving to a new place, and everything we use, it's basically fits in these small boxes. No special care required. This is just to emphasize that everything that you'll see today is publicly available data via data miners. Now, for us, it started about 13 years ago when I was a postdoc in an organ transplant lab, where as a electronics and communications engineer turned software engineer, looking at how biomedical and translational research is done, it felt like there was a fundamental flaw. And I want to emphasize that flaw does not mean that something's incorrect or wrong. It just means that there is an opportunity to fix the flaw while keeping everything else as is. And this flaw was, from a data science perspective, was the way the underlying philosophy in biomedical or biological research, where the goal always is to reduce heterogeneity. And there are three broad categories of heterogeneity when I talk about it today. Biological heterogeneity, which is usually age, sex, environmental exposures, and so on. Clinical heterogeneity, which is the type of disease, stage of the disease, treatment protocol that a person is on, So that would the way the diagnostic criteria that are used for a disease, the disease duration, early versus the late stage disease. So those would be the clinical heterogeneity. And then technical heterogeneity, which is everybody has their favorite technology that they like to use, that they believe in. And some people like, for example, Affymetrix microarray, some like Agilent microarray. Nowadays, we use RNA-seq with different library prep. So all of that introduces technical heterogeneity. And when we are carrying out any biological or biomedical experiment, we go to extra length to reduce these three sources of heterogeneity. We make sure that the patients are roughly the same age, their age and sex match, as we say it. We make sure that their disease is comparable. We write lengthy SOPs, standard operating procedure documents, to make sure that there is as little technical variability as possible. We have a whole field of batch correction to make sure that the data, there are no batch effects and so on. And let's take it, assume for a minute that we are interested in developing a biomarker or identifying a biomarker for a given disease. Could be diagnostic, prognostic, it doesn't matter. Then when we enroll a group of patients that are comparable, and then the controls, whether the similar disease or healthy controls, and then the data is generated by making sure that there is no as little technical variability as possible. Now, the next thing we do when we are developing this diagnostic model or identifying biomarkers is this single cohort gets divided into training and test in machine learning language, or discovery and validation, where the testing or the validation component is left out, never used until the final model is decided. And all of the other training or the discovery cohort samples, we use them repeatedly, we iterate over it to make sure that we found something that works. So assume that we enrolled blue people. And when we apply this model to unseen data from the same cohort, which are also blue people, it usually works pretty well. But we all know that this does not capture the heterogeneity of the disease. Results do not generalize. And that is why we are always asked, can you replicate this in independent cohort? And then the simpler way to say it is, while we know that it works in blue people, we don't know if it would work in green people or not. And this leads to the valley of death. A lot of things, a majority of the things from lab do not translate into clinic, from bench to bedside. And for me, this broken bridge is a symbol of this unknown real world heterogeneity, where we don't know when the bridge is going to break because we don't know what's going to happen in the real world. So this is a qualitative argument. Let me make it more quantitative argument along the same lines. When we are doing a biomarker discovery or a drug target discovery, before we generate data, before we analyze this data, every variable, whether it's gene, protein, immune cell type, it doesn't matter. Whatever the modality that you are interested in, each variable has 50-50 odds in favor of being associated with that condition, because we don't know. Think of this as an null hypothesis, right? Then we do our favorite statistical analysis. We pick our favorite p-value thresholds. Let's say p-value 0.05. We can argue about whether that's right or not, but let's assume for a minute. And then when we try to replicate, what we don't realize is that while p-value 0.05 tells us that there is 5% chance of this happening at random, what we don't realize is that if we were to replicate, there is still about 30% chance that the results would not replicate. Even if we choose more stringent p-value, you need higher sample size, but you still have at p-value 0.01, there is more than 10% chance that the results would not replicate. Now, a simple way to fix this is instead of choosing from 50-50 odds, let's choose a set of variables for replication or validation from a pool of variables that have 90% odds in favor of being associated with that condition. If we simply choose from this a diagnostic model with say p-value 0.05 and then try to replicate it, now we have less than 5% chance of results not replicating. So what we need to do is go from selecting from a set of variables with 50-50 odds to 90-10 odds in favor. And this is where the flaw exists, and the unique opportunity to really fix this flaw, and that is to embrace heterogeneity in this early phase of biomedical or translational research. And that's where publicly available data comes in. That's where data reuse comes in. As one of my friends tells me very dearly, that I'm cheap, lazy, and parasitic for using publicly available data that's available for free. And I tell him that's not it. The reason I like publicly available data is because I'm frugal, efficient, and symbiont. But the real reason I like publicly available data is because of not just the sheer amount of data that's available, but also because of the three sources of heterogeneity: biological, clinical, and technical heterogeneity are really represented in these data sets collectively. What do I mean by that? So think about pick a disease that you are interested in, a favorite disease of yours, be it autoimmune disease, cancer, infectious disease, doesn't matter. A lot of people across the world, a lot of researchers across the world are working on that disease. They each generate that data, but because in science we value novelty over replication, if somebody published a paper where the experiment is exactly what you were doing, we somehow try to figure out how it is different from this already published cohort or a published data set. We change it slightly, we change the age, we change the treatment, we change the disease stage, and so on. It is done, these cohorts come from different countries. So both the biological and clinical heterogeneity is really very well represented across all of these data sets collectively, not individually, but collectively. And different research groups use different technologies, different countries have different treatment protocols because different drugs are approved for use in patients and so on. So that also introduces this technical variability. And each experiment, each group works independent of the other one. So there is a lot of technical variability as well. So collectively, we have all sources of heterogeneity represented in these data sets. But the question becomes, how do you really use all of this data in single analysis? Because if we could now have a blue and a green people in the same analysis upfront, we know that not only it would be more generalizable or it would work in both blue and green people, but the a priori odds have also increased that it would work in unseen patient populations that's represented by like the purple, the yellow, and the orange people, right? So the challenge really comes back to there is so much heterogeneity that how would you really integrate all of this data? This is dirty data, as somebody told me a few years ago. And that's what our research has been, research in my lab has been focused on for the last decade or so, where we've now built a framework that is an associated R packages, a set of R packages that you can use from CRAN. But summarizing all of that in a single slide, everything that you will see today, remember that it comes from publicly available data. We did not generate a single data point ourselves. My lab doesn't own a single pipette. Once we pick a disease that we are interested in, we go in NCBI Gene Expression Omnibus or AMBL's ArrayExpress or any other data sources like import from that's funded by NIAID. So we go to these public data repositories and just look for data sets for the disease that we are interested in. Once we find the data set, we make sure that they are normalized. Each data set is normalized within itself. We never normalize across data sets. And we make sure that some basic statistical assumptions for that data type satisfied. So for example, in case of gene expression using microarray data, we make sure that it's locked to transform. Once we have identified these data sets, we divide them into discovery and validation, or training and test, like the way we do it usually. But the biggest difference is we never divide each data set into both discovery and validation. Each data set is either in discovery or in validation. So we want to make sure that our training or discovery analysis gets to see as much heterogeneity as possible. And we found that usually 4 to 5 data sets coming from different countries, different research groups, different technologies, collectively really represent all the heterogeneity that you would want to see in your data. Just to make sure that we never fool ourselves, we also sort of try to divide data sets by the research group or the principal investigator, just to make sure that there is no overlap. The same data samples are not used in both training and validation, right? So that's our central guiding principle. Same group, either all data from that group goes in discovery or in validation. Discovery, as I said, we try to select data sets so that as much heterogeneity is represented as we can. You need 4 to 5 data sets. These are just guidelines on how to select the variables of your interest, but feel free to change it. These are just guidelines, not rules. The R package is called Meta Integrator. That allows you to do the analysis. At its core, it's a random effects inverse variance model based meta-analysis, but some of the fundamental rules of meta-analysis are sort of ignored. So we, instead of making sure that the data sets are comparable to each other, we make sure that they are as different from each other. And what comes out of Meta Integrator is this set of variables that have 90% odds in favor of being associated with that disease or condition that we are interested in. We can now use these set of genes or cell types or proteins, apply machine learning that is best suited for that data type or the kind of question that we are asking. So it ranges anywhere from difference of means to deep learning and anything in between. So logistic regression, support vector machine, random forest, pick your favorite machine learning algorithm. We basically built a pipeline in the lab that once the variables are selected, you can just run through all of these models simultaneously with appropriate hyperparameter tuning to figure out what works. Once that is done, we log those models if it is biomarker. And then in unseen data sets, we show that these genes, these models can be diagnostic, prognostic, there are new drug targets, we are identifying new mechanisms that are drug targets, and so on. And I'm going to show you a few examples. Now, one question that I almost always get asked is, given how much heterogeneity you are looking for during your discovery process, how do you know that you have sufficient statistical power? So here is an example. Turns out if you have about 3 data sets and 150 samples, and you can see these data sets are not large, these are moderate or even small data sets. Each one is about 50 samples total in each data set. You still have 80% statistical power for detecting 2-fold change in expression or whatever the modality is. I'm just going to stick with gene expression from now on, but substitute for your favorite modality at p-value 0.05, even when there is high between data set heterogeneity. If there is no heterogeneity between data set for that variable, you can actually detect about 40% change in that variable. If you increase from 3 data sets to 4 data sets and 200 samples, now you have way more than 80% statistical power to detect even less than 2-fold change in the presence of high heterogeneity. If low heterogeneity or no heterogeneity, it's even more. So that addresses the statistical power problem. And then the question comes out, does this really work? Is there one example? And I present to you more than one example where we've really applied it, starting from organ transplant on the upper left corner to multiple infectious diseases, including sepsis, viral infection, tuberculosis, fibrosis, autoimmune diseases, vaccine response, and neurodegenerative diseases, cancer. We've really applied it to broad spectrum of diseases just to show that this framework, this set of guidelines and the associated code really is generalizable. A few things to remember on this, to take away from this slide is every single data point publicly available. We did not generate a single data point ourselves. That's number one. Number two, it's all human data, clinical samples from real patients, no model organism. This is the patient population that we are targeting, and the samples are coming from them. The total number of samples that were used in any given analysis ranges from 500 to 7,500, spread over anywhere from 7 to 40 independent data sets from all 6 inhabited continents. And each one of these projects, we started out as trying to say that what is the robust gene signature or a cell type that distinguishes the condition of our interest from everything else, either healthy or other conditions that it gets confounded with. And in each case, not only we found a diagnostic, but when we collaborated with clinicians and enrolled patients and tried to design studies to see whether we could have predicted this condition or not, in every single one of those, we were able to show that these diagnostic markers, locked diagnostic models, are actually also able to predict that condition before a clinician can in a real world setting. The only condition where you see the word prior doesn't appear is on the second one in the bottom row, and a group from University College London just showed that these 7 gene signature for distinguishing bacterial versus viral infection consistently predicted or diagnosed somebody with COVID-19, so correctly identified them as having viral infection 1 week before the nasal swab would become positive. These were healthcare workers that were followed every week as they came into work to make sure that they were not infected. And at 40% sensitivity, it picked up which healthcare worker actually had asymptomatic COVID-19 1 week before the nasal swab RT-PCR would show that it's positive. So that's the power that you can get from leveraging this heterogeneity, where you are identifying really I would be slightly bold to say that they are really the mechanism adjacent signatures for a given disease. Do these things really work, and what can you do with it? So not just you can develop diagnostics, but you can also start learning new mechanisms. And one of the biggest insights we've had is that host response to infectious diseases is really very modular, that can be used to develop clinically usable diagnostics that can diagnose presence, type, and severity of infection. So every signature that you see on the screen in a black circle has been independently validated by us or some other group at arm's length. Every single one except the 9 gene signature has been or is being translated into a point of care diagnostic by the industry, and those are highlighted with the company symbols here. But what you can now imagine doing this is it's not just for infectious diseases. You can easily imagine that we should be able to do this across a broad spectrum of diseases. Data already exists. So for example, if you ask a question that now that you have this diagnostic signature for viral infections or bacterial infections, how do you know that it is truly unique to that viral or that class of disease, and it does not show up in any other disease? How specific you can be? And the answer is you can really leverage this data that already exists, and you can show that the immune response to viral infections, the one that you see on the upper right side in orange, it's really unique compared to all the other diseases. And you can leverage and say, what is this cluster defining set of variables, and how do I use that in clinic? Another for example I would show you here, without going into too much detail, is, for example, you can see if you look in peripheral blood samples from patients with psychiatric disorders, depression, bipolar, schizophrenia, they still form their own cluster compared to all the other diseases. That's the power of being able to leverage heterogeneity across the large amount of data that exists in these public repositories, that you can start thinking about how to use them in clinic. Now, the problem in today's approach is, right now we feel like this miner. We are basically going into this dark tunnel where we don't know what data exists, what format they are in, and we are just, each book represents a data set for us, and we are just digging, looking at each data set, opening up, downloading, reading the annotations, the paper, figuring out if it's the right data set for the disease that we are interested in or not, putting it in our database, and then analyzing it. But what we really want to be, where we really want to be, is a library like this, where we can walk in, data sets are annotated, organized, that allows us to say these are cellular phenotyping experiments, these are transcriptome, epigenetic, GWAS, health records, that we can, if we can simply have this kind of a catalog, it would allow us to really, instead of having to do this manually for every single disease that we are working on, we would be able to turn the crank much faster. And that's one of the goal, or one of the things that we are working with Mark Musen at Stanford, who's also my division chief, where the goal really through CEDAR is to build these templates and build a metadata repository to enable these fair, compliant catalog of data sets in order to turn the crank for accelerating clinical translation. Now, I showed you a lot of data and said that you can do this, but where is the proof that this really does accelerate clinical translation? So we are in the middle of a pandemic, but there has been another pandemic that's been going on for hundreds of years, and that's TB, where the goal, about 1.5 million people die every year, 10 million people get this disease every year, 2 million people have latent TB infection. And in 2014, WHO set the goal that we want to end TB by 2030, and we need a triage test that will have 90% sensitivity and 70% specificity to screen patient population or general population, not just incoming patients who show up in clinic, but general population with 90% sensitivity, 70% specificity. And if we can have a test, a non-sputum, so a blood urine-based test, then we can end TB by 2030. So in 2016, we described a 3-gene whole blood-based diagnostic signature and showed that this 3-gene signature, 2 upregulated genes, 1 downregulated genes, across cohorts from 11 different countries, could distinguish patients with active TB from other respiratory diseases, latent TB infection, and healthy controls with the target product profile that met the WHO requirement. In 2017, Cepheid licensed this signature for clinical translation. In 2018, by collaborating with other researchers and data that was published in those next 2 years, we showed that this 3 gene signature that was designed for diagnostic actually could predict progression from latent to active TB 6 months before with an AUROC 0.86. So that again met the WHO target product profile for predicting progression. Now remember, 6 months ago, meaning patient has no symptom, they are not even coughing, no weight loss, no blood in sputum, none of those symptoms. And we are still able to diagnose that they are on their way to have active TB. Then we did an active triage screening, screening where instead of waiting for somebody to walk into the clinic with the symptom, we went out in the community and screened people, measured using RT-PCR, and showed that we could actually diagnose who has active TB but is not yet showing symptom in a real world scenario and meets the WHO target product profile. And then the same 3 gene signature could also correlated with a lung inflammation and identified those patients with active TB who would need a longer treatment. So what I just showed you is a single 3 gene signature that turns on 6 months before a patient shows symptom and shows up in a clinic. You could now go out and screen the community and use the same 3 gene signature for monitoring treatment response. In 2019, Cepheid gave me the first cartridge. This is a picture of a real cartridge that measures these 3 genes with a finger stick. You could use the small amount of blood, drop it in the cartridge, and 45 minutes later, it gives you an answer whether the patient has TB or not. And now we have so far published 3 studies. When I say we, it's not really my primary work. I'm part of the work, but it's Cepheid and Foundation for Innovative New Diagnostic that have published this cartridge and showed that the cartridge, using the same model that we built using gene expression data from publicly available data, still continues to perform, meets the WHO target product profile, overall AUROC 0.94. So when you get rid of all the technical and variability, performance, which was when we published was somewhere around 0.88 in terms of AUROC, has gone up to 0.94. So the point I'm trying to make is when you are using this heterogeneity, the performance, the accuracy estimates that you get that across all of this heterogeneity is a lower bound on how good it's going to be. We've seen this happen over and over again. Once you get rid of one source of variability, the accuracy tends to increase. And we've gone from a first description to an actual point of care diagnostic test in 3 years. That's the power in acceleration. We've done the same thing for drug targets as well in cancer. A friend of mine asked me that I'm interested in these 5 DNA methyltransferases, which one should I look at? And it has to be in KRAS mutation associated cancers. So he told me that it's the pancreatic cancer and lung cancer where you have a lot of KRAS mutations. So it took 1,000 samples of pancreatic cancer biopsies, 2,200 biopsies of non-small cell lung cancer, and SMID3 was always, the effect size was through the roof across these 3,300, almost 3,300 samples. So we knocked it down in a mouse model, and the picture speaks for itself that when you knock down SMID3, you see the tumors shrinking. Same analysis, but there was one gene that had 3 papers in PubMed called pseudogene or orphan receptor tyrosine kinase. There's nothing known about it in non-small cell lung cancer. In a mouse xenograft model, we showed that with the protein increases with as the cancer progresses, we knocked it down using siRNAs and showed that indeed the tumor shrink. And 3 years later, Pfizer published a trial targeting PTK7 in non-small cell lung cancer to show that it's safe, it works, and they are moving to phase 2 trials. So that's the acceleration, again, that you can get from using publicly available data. Another thing you could do, for example, is use you can ask new questions. So when people say that people who use publicly available data are research parasites, I say we are not research parasites. We are data parasites. We are not using or stealing somebody else's research productivity. We are symbionts, and we are really asking questions that we are interested in. And because these data already exist, we can really start asking novel questions and actually accelerate parts of the discovery pipeline. So for example, here we asked a question, can we find a host-directed therapy that would work across multiple different viruses? So we went to PubChem, we identified a bunch of small molecule screening libraries across 9 different viruses. From these compounds, we selected 30 compounds, or we have found 30 compounds that were not toxic, meaning if you just treat healthy cells, they don't die. They were effective against at least 2 viruses, according to PubChem data. And there was a transcriptome data available in LINCS, Library of Integrated Network and Cellular Systems. We used 5 of those as baits. The other 25 were used as positive controls for the results that come out. And then we had published a paper a few years ago showing that the viruses have a conserved host response. And we simply asked a question, find me drugs that have a transcription profile that look like these bait drugs, as well as the host response. And whatever drugs that we found, or small molecules we found from LINCS, in collaboration with Shri Group, we did quick in vitro validation to see whether any of those drugs actually work. Out of the 25 drugs that we used as positive control, this simple method produced 14 of those. That's number one. Number two, we tried about 22 of our first round prediction, and one of the drug first in vitro and using dengue viral infection showed that it actually did treat dengue viral infection in vitro. Therapeutic index was really large, 222. When we treat mouse infected with a lethal dose of dengue, this BCL2 inhibitor reduced the viral load, extended the survival. Less than half the mice died, and all of the mice that survived actually gained weight over the next 2 weeks as well. What is more important is if you look at the target of this BCL2 inhibitor in influenza challenge study, before they were challenged with influenza, so 24 hours before they get challenged, just the target of this BCL2 inhibitor actually predict who will get infected upon exposure to influenza. So that's, again, you can start using these publicly available data to ask novel questions and not just identify drug targets or molecules that could be repurposed as a broad spectrum antiviral, but they could also be used to find associated companion diagnostic to say which patients are more likely to be infected and would need this treatment. Another example that I want to give you, and then I'll stop, is you could also think about integrating the science of medicine, which is all the molecular data that are publicly available, and integrating it with the practice of medicine, meaning the electronic health records, to really accelerate clinical translation. And I'm going to give you one example where we were interested in idiopathic pulmonary fibrosis. 1 in 4 deaths are caused by fibrotic disease. The only curative therapy is lung transplant. Median survival is about somewhere around 3 years, and really a painful death. And we really need to find biomarkers for which patients are going to progress versus who's going to be fine. So we found 2 data sets, about 120 peripheral blood samples, gene expression profile in NCBI-GEO from patients with IPF. A method that we've developed in the lab called immunostates. We use this gene expression profile to just estimate proportion of 13 different immune cell types in silico, no collecting new samples and running them through FACS or CyTOF, and just estimated proportion of 13 different cell types in each of the samples. And what we found was proportion of monocyte was associated with long-term severe outcome. But this is a discovery cohort. We were looking for it. We found something. So what? So the next thing we did was monocytes are part of our complete blood count. It's measured across the world since mid-50s. We know how to measure it. You can measure it everywhere. So we went in Stanford Electronic Health Records and looked for every IPF patients who had a CBC count done within 30 days of its first diagnosis, and just looked at what the outcome of these patients were a few years later. And we chose a threshold of 0.95 kilocells per microliter because that's what Madeline Scott, the grad student in the lab, said her medical textbook said. So when we applied to Stanford IPF patients, indeed, it predicted which patients would have poor survival with a hazard ratio more than 2. We sent our code to Vanderbilt and said, could you run it on your cohort to see what the survival curve looks like? Still, they had a lot more samples, hazard ratio reduced, but still 50% increased chance if you have higher monocyte count at the time of diagnosis. And then we went to Optum through Population Health Sciences Center at Stanford, where we have data access to data through from 48 states, 5,700 IPF patients, and again, same threshold, 75% increased risk of reduced survival. Next, the question became, is this specific to lung fibrosis? What happens if you have any other organ fibrosis? So hypertrophic cardiomyopathy, systemic sclerosis, and myelofibrosis. Heart, usually skin, but other organs as well, and bone marrow fibrosis. Same threshold across all electronic health record data sets that we had access to, almost identical hazard ratio. This is a marker that every clinic in the entire world uses, and now we can start using it. We can repurpose that marker at the time of diagnostics to say which patients are more likely to progress and should be cared for. The other thing to think about is, imagine the way the disease was defined. It was not confounded by that. Different cohorts used different definition of fibrotic disease, and yet the same threshold consistently predicted that we can identify which patients are going to have a reduced survival. But this is, again, you can say this is, we could have just iterated over it so many times that we found something that works. 2 independent groups did the same thing independent of us, using the same threshold. And this is, one is in Australia, the other one is in UK, so completely different countries, very well characterized cohorts using the same threshold to say, yes, indeed, higher monocyte count at the time of diagnosis means reduced survival a few years later. We are doing the same thing by generating hypotheses and predicting which drug, FDA-approved drug, would work for a given disease, and then going into electronic health records to show that the drug is actually working for that disease as we expect. So in case of organ transplant, we showed that statin would reduce graft infiltrating cells, reduce injury, and extend graft survival. Electronic health record showed exactly the same. This paper comes out next Thursday in JAMA, showing that in ulcerative colitis, statins, if ulcerative colitis patients are on statin, the rate of admission, hospital admission goes down, colectomy rates go down, and corticosteroid treatment or the prescriptions go down. So I'll leave with the question that we've been discussing, data are not reproducible. Is that really true? I don't think that's true, because if data were irreproducible, we couldn't have done what we have done through these years. What is really going on, at least in my mind, is that the interpretations based on this data are not reproducible. So I'll stop here by just summarizing that while single study cohort, we need to continue doing them, we also need to recognize that they do not represent the real world heterogeneity. Real world heterogeneity comes from combining across data sets, and that increases our probability of success, especially when we are doing discovery or training analysis. We really need to think about heterogeneity upfront instead of trying to make data as clean as possible. And we cannot do this without data sharing. We have to promote it as much as possible. And I'll stop here by really thanking the people in the lab who do all the work, allow me to take credit for their work, all the collaborators who actually believe our data, enroll patients, and carry out these trials, funders who support our work, and most importantly, investigators who are sharing that data so that we can ask new questions and really accelerate clinical translation. I'll stop here. Thank you for your attention and take any question you may have.
Thanks, Purvesh, for the fantastic talk and grounding us with some real life examples on the value of data sharing and how it can be leveraged to kind of answer very specific questions. And also it's great, thanks for calling out the silent pandemic that's going on in TB and comparing it. And I recently remember seeing some staggering statistics about how many years it's been since the pathogen itself has been discovered. Goes back to 1892 or something like that. We have one vaccine that kind of works in children. And as of last year, I think the statistics still show that there are almost as many deaths as we've had with COVID-19. So I think it kind of is very profound, and I think emphasizes the fact that I think the importance of sharing data and being able to leverage it and reuse it. I see several questions over here, and we do have a few minutes. I think the highest scoring question so far is, can you say more about how we are leveraging CDER to improve data annotations?
Right. So it's still an ongoing work, and that's why I didn't have a lot of details. But what we've been able to do in the lab is we have actually built a database of at least somewhere between 50 to 70,000 manually curated samples. So that serves as our gold standard. And now we are basically building tools to automate, and when I say we, it's really Mark Newson's group, and we are just here supporting the fantastic work that Mark's group is doing, is using the tools developed in Mark's lab to annotate, for example, gene expression omnibus, and getting those annotations and comparing those against our gold standard manual annotations to say, okay, how good a quality that we are getting. And my personal choice is about 85% accuracy in this automated annotation would be really transformational. And once these annotations are done, essentially building a metadata repository in a CDER format that the vision really is can be made publicly available for people to query directly and know that what kind of data set for which disease in which country for which age group in which tissue is available, so that when you get the data set ID, you can directly query either NCBI gene expression omnibus, import, what have you, to download data automatically. You have these annotations, making your data more findable. It's already accessible, but making it more findable, and then allow you to quickly reuse using the APIs that already exist. So that's sort of the vision that we are working towards.
Great. So just my add-on to that, where does the annotation sit right now while this project is in progress in your current analysis that you showed? Is it just within?
So our gold standard annotations are in our server. They are metasignature.stanford.edu, where we've everything pre-processed, including gene expression data. So if you are interested in a given gene across all diseases, you could just go to Metasignature and look up. So right now it all sits locally, which actually is in some sense a copy of what's on Gene Expression Omnibus, or in supplementary tables of those papers. So when we annotate, we are also annotating through supplementary tables and all the obscure sources. But goal really is to provide as much of it as possible in CEDAR templates for everybody to use.
So a related question that's bumped up to the top is how much work has to be done to harmonize the metadata and make sure samples and the data in the repositories are actually what they're supposed to be?
So I would, it's a two-part question for me, although it's a one part, for me, it's two parts. So the second part is data in the repository are actually what they are supposed to be. Now, that's why I am saying that what we need is 85% accuracy. We've seen repeatedly in many of the studies that we published that there is invariably somebody, there is always one group, right, that didn't annotate that data correctly or the information is missing. But the power really is, without asking the original authors, we've actually been able to correctly predict what the right annotations are and then confirm with the original authors. So we need to make sure that what the paper says, so for example, sometimes there are disease severities that are in supplementary table but not really in geo records, or there are treatment information. The idea that we only provide group level statistic and not a sample level statistic in public data, we need to really address that as a community and say, if you have a figure in the paper or a table in the paper showing this data, you should be putting it in geo records. The argument about data anonymization doesn't hold there, because all data is already public. You are if you can put gene expression and all the other information out there, there is no reason why you can't put treatment data out there. So we should really be enforcing that. And I don't know how to do that, but that's what we need to do that. And there was a second question, but the second part, but the question's gone from the.
I know, I'm trying to find it too.
Yeah, so I'm sorry.
So let me move on to the next question in the interest of time, and I'll bring up the other question. So I think the next one is, what one thing would you recommend to data generators to consider while sharing to make such discovery easier for open data? So
I do this exercise in my class, the class that I teach on computational systems immunology at Stanford, where the homework assignments are very simple. I give them publicly available data, the paper that is published, and I say, go reproduce figure X. That's it. That's all they are supposed to do. And the idea is to really show them that, you see, when you assume that somebody else has access to that data, they don't have access to those data. So and then the lesson that the students learn is the data that they need to make public is every data point that would be required to recreate that figure. As long as you do that, we are good to go. So when you are sharing data, just imagine if somebody were to read this paper from scratch, and they were reading the figures and they wanted to recreate this, what would they need? You don't need to give your data. It would be good to source code. It would be good if you could, but if you don't, that's still fine. Just make sure that the figures can be reproduced, so that, and we are not out to show that somebody's conclusions were wrong, right? That's a waste of time. We would never get funding. Our goal, people like me, is to really ask new questions by integrating all of this data set. So feel free to share data. And another thing that I do in my lab to incentivize data sharing is just make them author. It doesn't hurt you. Make them co-author, get the data, and then make it public. Make their life easy, and things would move forward much faster.
Thanks, Purvesh. So I think we're at time, so I'll pass it back on to Susan for any closing remarks.
I just want to thank you so much, Purvesh. This has been a really enlightening and fantastic talk with a lot of really good questions and discussions, and I'm sure that folks who are attending who would like to follow up, certainly if you're interested, perhaps they could contact you. And I'm looking forward to learning more about your work as it progresses. It was wonderful. And thank you, Ishwar, for hosting the seminar today. Again, as a reminder, we'll have our October Data Science and Sharing Seminar, I believe, on the 15th. And I hope that you will all join us as we will post this recording and future recordings on our website listed in the chat. And with that, I'm going to close the seminar. And again, thank you so much.
Thank you.